Once again, welcome to everyone. I already recognize a whole lot of familiar faces. And I want to say hello to a lot of people I know. I see Chris Sander, Peter Rice, and a lot of other people that I've known over the years, as well as some new people that I haven't met. Welcome to everyone. This is the third of these coffees with EMBL. So I know I have my coffee, even in an EMBL mug, just to make it official. And these chats have become, I think, very valuable and very useful as a way of finding out what's going on around the world in this situation we're all experiencing. I think for most of us, the only time in our lives we've experienced this. And it raised a lot of issues. And so we just thought, just as a way, partly in testing that out, Peter has prepared, I think, a format to allow you to participate. And so we wanted to ask you to let us know in which decade did you attend or work at EMBL. And there will be choices displayed on the screen, which you can then select. So hopefully you can all see that, and then we can just have this interactive poll. So we've got a pretty good spread. Not surprisingly, it's slightly fewer from 1970 to 79 or from 2020, but in between there, we clearly have people representing, I guess, overall 6 decades of EMBL. So that's fantastic. And again, very inclusive, fantastic. One of the other questions that I think is going to segue very nicely into the next 2 talks we'll have is the interesting question that we've discussed at a couple of these events before is, as governments around all our countries keep saying that they are following policy based on science, an interesting question which we want to explore today is, do we think as a scientific community that the net effect at the end of this epidemic will have been to raise the profile of science or the reverse? Will the scientists get the blame for things not having gone well, or will our reputation have been enhanced and valued by society? Okay, so that's great. People are voting. You can clearly see that. I think we have, looking on screen, there seems to be a clear majority, about 7-1, in favour of the net effect being to enhance the reputation of science. And that's interesting. I'm sure that will trigger Graham Tebb, who's going to be talking later on, to respond to that and perhaps discuss some of the other possibilities. But I like to be an optimist. My cup is half full, actually slightly more than half full. So I think we're going to end up enhancing it if we communicate it correctly. And so the second half of today's discussion, we're going to be talking about how the science is perceived and how the epidemic is being communicated from scientists to politicians and to the public. And we'll have 2 talks around that. But we're going to start off today with 2 talks exploring the biology of the virus. And so I want to start off by inviting Peter Rottier to be our first speaker. And now, there you are, Peter. Yeah, I see you.
Great. Yeah, thank you, Angus. Yes, my name is Peter Rottier, as you can see on the screen, if it works all fine. I'm an emeritus professor at the University of Utrecht. I worked at EMBL in the early 80s with Graham Warren, had a great time, very inspiring. And actually, we had our first couple of publications together with Graham, one in Nature, one in Nucleic Acids Research, which was a popular journal at the time. And so today I have assembled some slides as a broad introduction. For some of you, this might not be so much necessary, but last time I had the impression that many people were not too familiar with coronaviruses, so I will present this brief overview. It will be give some general features, go into the virion, the infection cycle, and more specifically into the way the virus enters cell, which is an important phase in the infection where people try to intervene, and also into the accessory genes. All right. Let me see. I want to shift. How to shift to the next slide? Yes. Okay. So coronaviruses consist of a big family of viruses, which initially were imported mostly in the context of the veterinary scene. And there on this slide, you see a number of coronaviruses that were and still are important in the field, in the veterinary field, like viruses in felines, in canines, in chickens, in pigs. And say before the first SARS coronavirus outbreak in 2002, there were 4 human coronaviruses known, and all of these were just causing mild common colds. So they were not considered very important. By hindsight, from what we know now, but not at the time, we know that most of these, probably all of these human coronaviruses came from zoonotic events. We know that quite sure for the one at the top, human coronavirus OC43, which about 100 years ago jumped from cattle to humans, and that was historically, you can find back that there was a pandemic of bronchiolitis, so also quite severe symptoms, respiratory symptoms, as we know them nowadays. So already 100 years ago. So later on, we met with the SARS coronavirus in 2002, which started in southern China and traveled to Hong Kong, from where it spread to all over the world. As you remember, it was a virus that was coming from bats, but used civet cats most likely as an intermediate host. And then 10 years later, approximately, we had the MERS coronavirus that popped up in Saudi Arabia and was using dromedary camels as an intermediate host. And nowadays, of course, we have the SARS coronavirus 2. This is just an overview to show that these coronaviruses can be distinguished in different groups, different genera, alpha, beta, gamma, delta. And if you look in the middle, you see that they occur in many species. And in some species, you have multiple different coronaviruses. Like in pigs, you have at least 4. Also, the one at the bottom, the Delta coronavirus, which occurs in pigs. So these are quite multiple, and what they generally do is cause enteric and respiratory infections, which evolutionary is understandable, because those are the ways by which the viruses can spread and maintain themselves in the ecosystem through coughing, sneezing, and by the feces. They phylogenetically, they can be grouped together, and you find the alpha coronaviruses here in yellow, the alpha coronaviruses to which the 229E human coronavirus and NL63 human coronaviruses belong. And then you have here on the left-hand side different clades of beta coronaviruses, to which, for instance, MERS coronavirus here in this clade, SARS and SARS-like coronavirus in this clade, and here you have human coronavirus OC43 and mouse hepatitis virus, which I mentioned because it's the type member and was used to study coronaviruses fundamentally in most cases, certainly early. Okay, a little bit about the viral particle. It's coronaviruses are enveloped, which means that they have a a membrane envelope, and they contain a positive strand RNA, which is the genome. And the genome size is extremely large. These are the largest messenger RNA-like RNAs which occur in nature. And if you look at the picture down here, the schematic drawing, which is which summarizes what we know of these viral particles. On the left hand, you see the typical EM structure of these particles. So in the viral particle, you have the RNA genome, which is packaged by one particular protein, multiple copies of that, to form a helical nucleocapsid, which is inside of a particle that is covered then by a lipid bilayer. And in this lipid bilayer, we find 3, sometimes 4 membrane proteins. The most conspicuous one is the spike protein, about which we will hear more. Then we have the M protein here, a triple spanning membrane protein it is. And then we have the E protein, which is an envelope, which is called envelope protein, but and that's what the E points at. So then below we see the genome structure. So this is the RNA with the 5' and a 3', typical messenger RNA, but very long. And about 2/3 of that RNA encodes the polymerase functions. It encodes a pol-1A and a pol-1AB, which are precursors that are subsequently cleaved into a total of 16 functional proteins. Downstream of the polymerase, we have the genes encoding the structural proteins. So we see there the S, the E, the M, and the N proteins, which are part of the viral particle. And in between those, you see, for this case, which is for a feline coronavirus, you see genes 3A, 3B, 3C, and 7A, 7B. And these genes are what we call accessory genes. They are in most cases, they can be deleted without the virus becoming non-viable, but they contribute specific functions, particularly in the pathogenesis. When we then look into a summary of the replication cycle of these viruses within a cell, so what you see is a diagram of a cell, and on top you see a virus particle binding to the cell by binding to a receptor, and then being endocytosed, and then the virus fuses its envelope, its membrane, with the envelope of the endosome, and that introduces then the viral RNA, the messenger RNA, into the cell. And this messenger RNA is seen by the cell as a normal messenger, and ribosomes start to translate it. And they translate from this long messenger only the pol-1A and pol-1AB part. So these 16 functional proteins that come from these precursors by cleavage, self-cleavage, most of them, they result in proteins that form structures in the cell that you can see here. We call them double membrane vesicles, and in which the virus hides to perform its transcription and replication. So we have there, on the one hand, replication within these structures, which means that the genomic RNA is by a polymerase that is generated, that is one of these 16 proteins that is generated from the pol-1AB domain. It makes a copy, a minus sense copy that you can see here. This full strand minus sense copy then can be copied again and produce numerous plus sense copies of the original infecting RNA. And then what in addition occurs is synthesis of smaller messenger RNAs, which we call subgenomic messenger RNAs, from which the the genes that are downstream of the polymerase, so the ones for the structural proteins, are then produced. And without going into the details, these messengers are then translated. They produce the spike, the envelope, and the membrane proteins, which are inserted into the endoplasmic reticulum, and which then are transported to the ERGIC, the ER to Golgi intermediate compartment, and that is the compartment where new viral particles are being assembled, because the full-size copies of the genomic RNA meet in the cytoplasmic the nucleocapsid proteins that are produced by one of these subgenomic RNAs, and they form this helical nucleocapsid, which docks to the membrane proteins, the viral membrane proteins that are accumulating in the ERGIC, and they then bud into the interior of the ERGIC, and this creates these vesicles which move to the Golgi and through the Golgi, and in the end the viral particle leaves the cell with fully mature glycoproteins, and the virus is then infectious and able to infect new cells. Okay, this is a picture just to show these double membrane vesicles, which these viruses use to hide and to in order not to signal to the cells that there are foreign invaders, because cells are educated to react with all kinds of substances like interferons to counter the infection. So viruses have evolved to try and avoid those reactions from the cell. So here a picture where we see the process of virus budding. Here on the left you see the start of a budding particle. Here the budding is a little bit further. The blackened thing is the membrane, and you see it increase. Here it is almost complete, and here we have complete particles which then further mature and are released from the cell. How many particles are produced? Well, many, and that's interesting to realize, because, well, you can see here for the SARS coronavirus, the SARS coronavirus 1, this is after 15 hours in vero cells, and you can see here these vesicles with multiple viral particles, but also viral particles already on the outside of the cell, some of which rebind to the same cell, and most of them detach and go for other cells. This is at a very late point in the infection on the right-hand side, and you can see that hundreds and probably per cell more than thousands of new virus particles are produced, which then, if it is in the respiratory or nasal epithelium area, they find multiple new cells, and you can imagine that within a couple of days, a whole sheet of epithelial cells, and these cells die and continue to produce new virus, and then ultimately die. So the force of the infection is enormous. Then some more about coronavirus entry. This is again a picture of in the middle of the coronavirion, and on the right-hand side I summarize again the entry pathway, with in yellow here the receptor, and this is then the endosome, etc. So it is the spike protein that is doing the entry job, and the spike protein is a normal type 1 membrane glycoprotein, which means it has a signal peptide at the N terminus, which is cleaved off, and it has a transmembrane domain at close to the C terminus, and after that a small endodomain. This is inside the viral particle, and the bigger part is outside of the viral particle. this spike protein can be distinct. In that protein, you can distinguish an S1 and an S2 part, as we call it. The S1 part is the domain, more or less the top domain of the spike that is involved in receptor binding. And the lower part, the S2 part, is involved in the act of membrane fusion. Okay. The structure of this. Yes,
Peter. I think just because time is so short,
right,
and I really wanted to give a chance for people to come by and have some questions. I know we can do some of that in the follow-on, but right. could we? Yeah, if you could just maybe quickly wrap up this part. Yeah.
Right. So I will. Well, the spike protein is doing its job. It has it brings together the viral membrane and the host membrane. The spike protein has been, the structure of it has been solved after many years. In 2016, we were the first to show the spike of the mouse hepatitis virus. And it's a really neat structure. If you can look at this, this is the surface view where you can see that the spike is a trimer. You see the three colors of the three molecules. They embrace each other very nicely, and you can see the different domains. You can see that in another type of view. And if you look at here in the middle, the top view, you can see that each spike falls into different substructures. And these substructures, domain A, domain B, for instance, they can function as the receptor binding domains. Here is one molecule here with the domain A and domain B at the left-hand side, and on the right-hand side, you can see that for mouse hepatitis virus, domain A is the receptor binding domain. For other viruses, like for the SARS coronavirus as well as MERS, domain B binds to the receptor. What are the receptors that we know nowadays? Well, they are indicated here because here you have these different domains. For the NL63 human coronavirus, it is ACE2. ACE2 is also used by the SARS coronavirus, as we know, both the first SARS coronavirus as well as the one that is circulating now. Then we have APN, which is used by coronavirus 229E, but also by a porcine coronavirus. And then we have here DPP4 used by the MERS coronavirus. All these receptors are function normally as enzymes in the respiratory tract, and that is because probably the viruses use them because they are very abundant in the respiratory and enteric tracts. Okay, then a few words about the other genes, the accessory genes. This is again for the some different viruses, the genomes. The accessory genes, I could say that in summary, those are genes that usually interfere with the innate immune response of the infected cells. So they are trained to reduce the immune reaction by the infecting cells. Well, then I think
Peter, I think we're going to have to move on just because of the incredible time pressure.
That's fine.
This is, I'm fascinated with this information. I'm sure, like myself, the audience has a large number of questions. And are you happy to take questions from the audience in the follow-up?
Sure, whenever. Maybe later. Maybe.
Yeah. Let's do it that way. And let's bring in Stephen.
Greetings from EMBL in Grenoble, where it's 31 degrees. It's summer before its time. I saw on the list that there were people alumni from Grenoble, and so many greetings to Valerie, I can see. And Ru Yan, who's in Shanghai, if she's there, and of course Andrew Miller, who is the first head of EMBL Grenoble. In fact, I joined the lab when he was head now many, many decades ago. So I've been here an awful long time, but I'm still here working on influenza virus mainly. So what I'm going to talk about to you today is, can we repurpose any anti-influenza drugs for use against the coronavirus? So can you go back? Yeah. So the next one. Yeah, this is what I'm going to kind of talk about quickly. I hope So influenza and SARS are pretty similar in some respects, but basically their molecular biology is very different. They are both envelope viruses. They both infect the respiratory tract, and in their severest form, they can both cause viral pneumonia and hyperinflammation of the lungs. Both are easily transmissible via droplets. Both have proven pandemic potential and potentially kill hundreds of thousands, if not millions, around the world. However, the molecular biology of the two viruses is completely different, except for the fact, and this is what I'm going to focus on, that they're both single-stranded RNA viruses, and therefore both possess an evolutionarily conserved RNA-dependent RNA polymerase that replicates the viral genome. And you heard a bit about that from Peter. Even so, they are quite different families of RNA viruses. Influenza virus is a negative sense virus, which means its genome is, and it's also segmented in 8 pieces. So the virus contains 8 pieces of negative sense genomic RNA, which means the viral polymerase has to first transcribe the genome before viral messenger RNA is available. Furthermore, influenza replicates in the infected cell nucleus. Coronaviruses have a positive strand RNA genome that you heard is unusually long. It's about more than twice the coding capacity of influenza. And coronaviruses replicate in the cytoplasm, and the positive sense means that genome can directly act as messenger RNA without being transcribed first. So my group has been studying influenza RNA-dependent RNA polymerase for the last 15 years or so. And this polymerase has many unique features, and our goal has been able to understand it mechanistically by structural biology. Our latest results using cryo-electron microscopy, where we were able to follow the complete cycle of transcription, were just recently published in Cell. In parallel, we've been using, and others, our structural information which has been underpinning development of the next generation of anti-influenza drugs that target directly the influenza polymerase. So let me just remind you in the next slide what a polymerase does. So the circle here represents the polymerase. The yellow is a sequence representing the viral genomic RNA, which we call the template. This binds into the active site of the polymerase, and then also binds what's called a primer, a short RNA sequence, which anneals to the end of the template. And then there's a binding site for an incoming NTP, in this case ATP, which is selected because it's complementary to the U in the active site. And then the next slide, skipping a few steps, what happens is that the template and the product subsequently translocate one nucleotide at a time as each incoming NTP is added to the product in a way that's complementary to the template. So like this, the polymerase chugs along the whole of the template, making a complementary copy of the other sense strand. So the question is, with influenza virus, has a very special mechanism for deriving this primer, the part that's in blue. Well, first of all, the structure of this was the structure of the polymerase that we determined about 6 years ago now. And here you see this yellow template end of the RNA going into the active site. One of the curiosities about flu is that it actually binds the complete other end of the genomic RNA as well, what's in pink there, and that activates the polymerase. But on top are shown these 2 extra domains called the cap binding domain and the endonuclease. The next slide, these are used in snatching capped RNAs from the host cell polymerase 2. I remind you, this is happening in the nucleus of the infected cell. The viral polymerase binds to the mammalian, the host pol2, which is churning out capped RNA transcripts. And these are bound by the cap binding domain of the viral polymerase and cleaved by its endonuclease, giving this red capped primer, which is then rotated into the active site to initiate transcription, as I showed you in the previous slide. So this is an incredibly special mechanism, but when we solved the structure of these domains, we could see that they would be ideal targets for antiviral drugs. So without going into a lot of details, over the last 10 years, 2 new classes of anti-influenza drugs have been developed. One is shown on the left here, exemplified by Remdesivir, developed by Vertex and Janssen, and this is a cap analog. So it binds and blocks the cap binding domain of the viral polymerase and stops it doing any cap snatching and therefore stops transcription. An endonuclease inhibitor, so this is the other domain which cleaves the primer off from the nascent transcript. And here we have Baloxavir, which is now marketed, actually. It's been on clinical use in USA and Japan since 1 year now. It's marketed under the name of Xofluza, and this potently blocks the endonuclease of the influenza virus. So these 2 drugs are real breakthroughs, if you like, in the influenza field. I'm telling you all this only to say that they're completely useless against coronaviruses, because this is an incredibly specific mechanism of influenza virus. And in fact, coronaviruses don't need to cap snatch anything because they have their own capping enzymes encoded in the genome. So now let's focus in a bit more closely. Here's a comparison of the structure of influenza polymerase, which we determined with the primer template RNA, and a incredibly recent structure just from a month ago of the SARS-CoV-2 polymerase, again with a primer template RNA. You can see they're different in size, shape, and everything when you look at it like this, but you'll see that the RNA, the red and the yellow strands are pretty much the same. And if we focus a bit closer on the next slide, you can see that the core of this enzyme is virtually identical. And that's because they're doing this very evolutionarily conserved mechanism of nucleotide addition, template-directed nucleotide addition. So this is the real core of the dependent RNA polymerase. So the question is, can you target this highly conserved region? And the answer is yes. So here is a drug that was developed initially against influenza. It's called favipiravir, or T705. And this is a class of drug called nucleoside analogs, which mimic ATP, GTP, CTP, or UTP and bind directly in the active site of the polymerase. And this particular one mimics actually ATP and GTP, and it functions what's called by lethal mutagenesis, which means that once it gets incorporated into the product strand, you're losing coding fidelity because the included nucleotide analog can mimic either an A and G and base pair with a U or C. So any subsequent copying of the genome strand will give rise to errors. Similarly, the next compound does the same kind of thing. It's a cytidine-uridine analog, which again causes lethal mutagenesis. Now, the first one was approved for use against pandemic influenza by the Japanese government and is now being resuscitated as an anti-SARS-CoV-2 drug. And as with all these new drugs, there are several that are being tried out. Some clinical trials are showing some effect, but overall, we've not got enough data to really claim any of them are working particularly well. The one at the bottom is not even in clinical development, but it works in vitro. Again, this was published very recently in Science Translational Medicine. And all this brings me to Remdesivir, because this is in the same class, and this is the one that everyone's talking about now. This was developed by Gilead to inhibit Ebola polymerase. And an Ebola virus is an RNA virus as well. It's a negative strand virus, but not quite the same family as influenza. Anyway, this drug again works by directly being incorporated into the product by the RNA polymerase. And then this works in a slightly different way. It's thought to be by what's called delayed chain termination.
In other words, it is an ATP analog that's incorporated opposite U, but it doesn't actually stop the polymerase until 2 or 3 more nucleotides have been incorporated. And then for steric reasons, it just seems to block translocation. Anyway, this, as I'm sure many of you are aware, has been in clinical trials. So it's now, there was some kind of positive results from a clinical trial in the USA recently showing it shortened the time it takes to recover from COVID-19 by a few days, and hence has been authorized for emergency use in the US and Japan. And one of the issues with this is there are side effects. It's a bit toxic. In fact, all these nucleotide analogs tend to be because they can interfere with other polymerases, cellular polymerases. But this one is also not generally in clinical use up till now, and therefore its availability is very low. It's an intravenous drug as well. And I read recently that Gilead was hoping they might have 1 million doses made by the end of the year. So with that, I'm going to finish again with a summary. So I've gone through all this. The recent flu drugs are very good against flu, but no use against COV2. But these other nucleoside analog drugs may be useful. They're more broad spectrum, but rigorous clinical trials are really needed to see how well they'll perform. So with that, I'll stop, and if there are any questions.
Thank you very much, Stephen. That's fantastic. I noticed a couple of things. One thing, my understanding from the recent media reports is Remdesivir shortened the duration of the illness for patients who recovered anyway, but didn't actually reduce the mortality rate or prevent the number of people who died. Is that correct?
That is correct for that clinical trial. Shortened from 14 to 11 days, something like that, which could be significant. But obviously none of these are a magic bullet yet. And I don't think any of the drugs available are going to be magic bullets. And we're just looking for something that helps a bit, because to get a new drug from zero on a drawing board is usually 10 years. And
I noticed you didn't talk about the clinical trial being carried out by President Trump on chloroquine on himself. What can one say? Okay, I guess you didn't fit that into your model here, but that's fair enough. So actually, just before we move on, and I think I really encourage we have. There's so many questions here. I think we'll have the questions in a little bit later. But obviously, there's a lot of people, a lot of governments, I think, putting a lot of money into vaccine development. And there's very mixed opinions, as I understand, about the prospect. And 2 weeks ago, Peter, perhaps you can correct me if I misquote you here, but you were saying that there have been no effective human vaccines for any of the coronaviruses. Is that correct?
Yes, that's still correct. Yes. There are no on the market except for the infectious bronchitis virus in chicken, which is a highly passaged virus that has been used as a vaccine. Not extremely successful, but it is still being used.
So that's very important. I wonder if we could just take a straw poll amongst everyone. Do you think, having listened to the two presentations, especially to Stephen's review, that any of the current existing antiviral drugs can be successfully repurposed for treating COVID? Yes or no? I'm going to vote no, but and we just like to collect your thoughts on this, or been influenced by it. And okay, so the figures I'm seeing at the moment still changing a little bit. It looks to be about 2 to 1 that people are thinking we can't rely on just simple repurposing, which isn't the same as designing a new drug informed by the structure, Stephen. I think that would be a fair thing to say, but that perhaps we won't be rescued by just transferring one of the existing drugs over, at least from the information available. Thank you for that. It looks to be 2 to 1 against it. And if I could then, if we could zero that again, Peter, and then just ask, are you confident that any of the current vaccine trials in development will deliver an effective human vaccine against SARS-CoV within the next 6 months?
Well, I do think so. I think the first results are out for the recombinant adenovirus vaccines, which are used by the Oxford, the Jenner lab, and by Johnson & Johnson. The experiments in rhesus macaques seem to be promising, although the latest result was that these the vaccinated animals still shed virus. So that would mean that the animals still could be, or the vaccinees could still spread the virus, but the effect was that the clinical signs were reduced by the vaccination. These are just one example of vaccines, and I think of a type where a recombinant approach is being used. There are many other approaches, for instance, approaches by which certain genes are deleted from the coronavirus itself, so you get a genetically attenuated vaccine virus. I would expect that such kind of vaccines could be successful, also from my own experience.
Yeah, I think that would be an excellent topic for us to take up and really focus one of these future discussion sessions on different vaccine strategies. And again, please, if people are interested in that, do write into us and let us know your opinions. But I think one of the things that is obvious to me in listening to the two excellent scientific reviews being presented here to a specialist scientific audience, and just in case anyone couldn't see it, we got a 50-50 response in terms of your enthusiasm or confidence that we were going to have a successful vaccine developed. So that's interesting. But I think one of the real difficulties that we're all experiencing is how this type of complex scientific information is communicated to a broader general public across all of our countries who are affected, including to many people who don't have the sort of scientific background that we in the meeting today have. So I'd like to bring in Carmen Fernandez. Carmen, are you there?
Yeah, first of all, I would like to introduce myself. Maybe people then will understand why I say what I say here. So I'm a scientist by origin. I made my postdoc at EMBL Heidelberg from 2009 to 2011. And then I came back to Madrid. Actually, I live here in Madrid since then. And somehow, I don't know, don't ask me, maybe we can leave this for the next coffee session. I decided to study a master's degree in science communication. So I started to work both in the lab, working in NMR and applying NMR to structural studies to study molecular interactions, and working on the communication of the work that we were doing in my institute in Madrid. So from that point of view, and also I've been working in radio and also in another media, writing in newspapers and so on. So arriving to this point with the pandemic, it's really interesting to me. I mean, maybe the word is not so nice. No, we are in a really dramatic health crisis. But we have to consider that we are actually also living a very interesting time in the sense that we are experiencing how the outbreak goes and how it proceeds minute by minute. So we are getting the information updated almost to the second when it's happening. So this is something to live and to consider for future pandemics. So science is always very difficult to bring to the media. All institutions, like I guess it happens maybe for EMBL or for us at the FSIC in Spain, you try to promote science, you try to reach the media with this content, but it's quite difficult that newspapers or even more television would buy it, let's say, no? Because you were telling before, Angus, how difficult it is to translate these these words, this difficult science, into general words that society or the general public or the citizens can understand. It's almost impossible how to explain them what a PCR is, or even DNA. For most of the people, they don't even know what is that. Imagine RNAs, that is the basis of this coronavirus. But now, you turn on the TV, you open the newspaper, and it looks like everything is science. You see, I don't know, somebody will do these studies and somebody will study the statistics of the number of scientists that now appear in the media. No, it's something amazing. So for one side, it's really good for science, the moment that we are living, because for us, it's a way to reach people and to explain to people what we are doing. And not only the work we do in the lab, but also that for me, it's a bit more important, and we will discuss later on over this, how science works. Because we are very used to the periods of time and how many time it takes for the things to come up or to get knowledge or to create knowledge. But the general public, they don't understand. So it's a good time.
So what one comment on this, Carmen, if I could just say, is that really for me underlined this problem in understanding is in the UK recently. There was a very unfortunate situation where a small subset of people believed misinformation in social media and in the public domain that the virus was being caused by 5G telephone masts.
Yeah.
And they were actually knocking down telephone masts and assaulting telephone engineers. And that really underlines the difficulty in communicating.
Yes, I mean, I was coming up to that point. Let me tell you a bit later. So if we think how research is developing also at this point, just think about the timelines of the coronavirus, these new coronaviruses. No, we heard about it in 31 of December last year. So it was when the WHO announced it that there was a kind of a strange disease, different pneumonia in China. Then the 10th of January, so 2 weeks after that, more or less, we had already the sequence. We had the genome, the full genome of the coronavirus. 13th of January, I think actually in Germany, the PCR kits for detection came up. So we are going really, really quickly for the standards of science. So we are actually moving faster than we would actually do. But it's not enough. People doesn't understand. People is frightened. It's something that is touching the health. So it's the most interesting topic for everybody. It's not enough. They want to have the drug in the hand already to take it.
Yes,
so it's really difficult.
So that's a really important point. And so I apologize. I'm just very conscious of the time. And I mean, one of the other things I was hoping you could comment on, and we could discuss. Of course, it isn't just some crazy people in the general population who are misinterpreting science or misunderstanding it. I have to say, I apologize for making a political comment. I have been horrified, for example, at watching some news conferences given by the current US president in terms of his representation of science and suggested treatments, which really concerns me. But if I don't want to go down that rabbit hole. I just want to say another problem in science communication is very few of our politicians who are making the decisions about whether we are forced to stay at home, whether our children can go to school, whether that we lose our jobs, or whether the factories close or the economy collapses. Very few of these politicians are scientifically literate.
Yeah,
and I simply mean by they don't have science degrees, they don't have formal education in science, and so it slightly worries me about how are they receiving information, from which scientists do they receive information, and how can they form policy if they don't understand it? Could you maybe comment on how the scientific community can better communicate with our political masters? Because I'm not sure how that's happening.
I can tell you, I know better Spain, of course. Maybe somebody else can explain later about their own countries. So here in Spain, it's really quite difficult to arrive to the politics. But in this case now, with the pandemic, our president, when he comes out, he always says that every decision that they are taking is taken under the advice of experts, so scientific experts. But on the other hand, we don't know who those experts are because they are denying to tell the names. So this is quite interesting somehow because you don't know who is the people that is really advising. I mean, you expect that they are scientists that are working there. Sometimes you see them in meetings, but it's not really clear what is happening. There is kind of obscure, relationship between them. I mean, it's very difficult to say now what is going to happen. But actually in Spain, it's like this. Also, we are... politics expect an answer that is yes or no, or do this or do that. And scientists work under uncertainties. We need data, we need evidences, and it's very difficult to reply and say, no, I don't know. And then this person would say, okay, if you don't know, then take whatever. Because
absolutely. The politicians have to come up with short-term answers.
Exactly. I mean, the point is that we work under uncertainties, and we really need time to be able to give answers. So all those gaps that are there in the middle are filled up, as you said before, with misinformation, disinformation, fake news. I mean, the virus was created in a lab in China or whatever. So it's kind of difficult to try to help with that. Also, because it's not only the mass media, it's also the social networks. We are all in Twitter or Facebook, and you all know how it works. At the end, the rigorous information that is also there gets in the middle of a bunch of misinformation, trolls, people telling whatever. And then it's really difficult to look for the right information also there. So for people, it's a really difficult exercise to reach the information. The information is there, but it's not easy to arrive to them. And
yes, it's diluted by too much information. Could I bring in Graham Tebb from Austria? Carmen was talking about how this can vary between different countries. Graham, could you comment on the situation that you've seen developing in Austria?
Well, there's a couple of points. Thanks, Angus. First of all, you asked us to introduce ourselves. So for those of you who are similarly old to me, I was in EMBL from 86 or 85 to 88. In fact, Angus, when you arrived, I was in the lab next to you and we had a lot of fun chatting away together. I still remember those days very fondly. And one of the things I really remember is your enduring optimism. And you've asked me here to play the bad cop, to try to dampen your optimism. 4 weeks ago, you were very positive about how much attention science was attracting in this current pandemic. You thought it was great that science was so much in the public eye. I agree with you. Even in my hockey club, which has thankfully reopened again, 2 meters distance, of course, people are talking about science. They're talking about the R values. They don't have a clue what it is, of course, but they understand that when it's bigger, it's bad, and when it's smaller, it's getting better. They don't understand the errors, the assumptions, anything, but they understand a number that's being touted in the media. My worry is that there is a huge need for information, and there's an information vacuum. And at the moment, scientists are being expected to fill this vacuum. Qualified scientists can't. And a big question I have is whether they should be going out further on a limb, saying things that aren't perhaps as rigorous as they would like to be, in the awareness that if they don't say things, other people will. Now, when I was young, sitting having my cornflakes in the morning with Radio 4 blaring in the background, the BBC had a science expert who would comment on all matters scientific. I think it was Steve Jones. He was a sort of poor man's version of an Angus, with a Welsh accent instead of a Scottish one. He was actually an expert in some form of snail that grows in Derbyshire. but that qualified him to talk about everything. And these people, Carmen calls them todólogos in Spanish, know everything, understand science, are vaguely scientifically literate, and they are filling in the information gap. Should we be allowing them to do that? Angus, you said that it's a real pity that politicians don't have a scientific background. When you and I were a little bit younger, we did actually have a prime minister in the UK who had a chemistry degree. I think she was the only one with a science background. And I gathered from our conversations that she wasn't exactly your favourite. So maybe a better understanding won't help. Carmen said that...
Yeah.
Carmen said...
Go on.
Sorry about that. Carmen said that in Spain it's not clear which scientists are advising the government. It's great that the governments are listening to scientists, but who are the scientists? Now, in Austria, we have a similar situation, except that we've now found out the scientific background for the fairly draconian measures that our government has introduced. A bit of background here. Current figures show that the new COVID has been given as the cause of death for 605 people in Austria since the start of the year, which is a fraction of the number of people who die of the flu every year. It's sad for every one of them, of course, but the median age of those who have died has been 84. I think there's only been 11 people below 65 who have died. It is primarily a disease that kills elderly people, and there will not be a noticeable increase in the overall mortality in Austria this year. I think we're all happy about that. I recognise that this is different in other countries. Austria is unusual in that it has a very good health system. I think at no stage were more than 20% of the ICU beds occupied by COVID patients. Hospitals are fine, they have plenty of capacity. But in the very early stages of the pandemic, a group of mathematicians whose primary expertise is in financial modelling, the stock market and so on. As an aside, if they were that good at it, they wouldn't be working as professors at the university, came up with a mathematical model that they sent to the ministry, promising 500,000 deaths within the next few months, complete breakdown of all our health services. As a result, the country shut down. We're now slowly opening up again. We have to go out like this. So if I'm outside, I don't have to wear this fetching pink number. As soon as I go indoors, I do. If I go into a restaurant, I wear a mask. I can take it off to eat, but I put it on again to go to the toilet. And this despite the fact that the cluster analysis in Austria has shown that there have been no cases of transmission in restaurants, no cases in schools. They were shut for 2 months. No cases in public transport, no cases in anything. So the facts that are now coming to light suggest that the measures we adopted were exaggerated. Now, I'm not and this backs up what Marco said 2 weeks ago. A very small fraction of people in his country, Italy, are being threatened. Everyone is suffering. When I said that there have only been 600 deaths caused by COVID, I have an awful feeling that I'm understating it dramatically, because the primary rule of medicine, I'm not a medic, is primum non nocere. Whatever you do, you should not cause harm. Now, what we have done is essentially close our hospitals to all non-COVID related things and all non-essential operations. So all the screening programs have been stopped, all routine operations have stopped, people have been locked in, there's been goodness knows how many increases in wife beating and so on. There have been diseases, I guess, going undiagnosed and thus untreated for a long time. I think the cure in our case, in our peculiar case, has been, I would say, worse than the disease. So the question I have is whether this blanket panic-inducing, you can't go out, you must stay in, you can't invite people over. Police were actually fining people for inviting friends to have dinner with them. All of those fines have been thrown out by the courts, by the way. But were these measures justified? So the two.
Yes, so I think that is a really important part of an argument, and something. Thank you, Graham. Can I just say, by the way, in my defence of optimism, if you grow up in Scotland with the weather we have, you have to be an optimist. That's my excuse for being optimistic. Some people were commenting as well. I was catching on the chat. I mean, clearly some politicians do have some scientific background. It's a very small sample size, but Angela Merkel, of course, is perhaps the most prominent one who was trained as with a scientific background. And I think objectively looking, we have to say Germany is one of the countries that's coped with the crisis better than most. And congratulations to them for managing that for whatever reason. I think scientifically, my own feeling is we'll do a good job as scientists at some point in the future when we can look backwards and then we have more detailed, comprehensive data sets from all the countries and a much clearer point of view. It's very hard for science to comment authoritatively without having enough data to draw your conclusion, because of course how science proceeds, as we all know, is by gathering data, forming hypotheses, testing them, and then refining and changing them. And unlike politicians, we are prepared to revise our opinions based on data that we're presented with, rather than doggedly defending a previous point of view. But I think how science is presented in the media, I actually, despite being an optimist, I have a worry that the scientists are going to be scapegoats for the politicians to say, well, we're only doing what the scientists told us. And yet an important question is, how does information flow from the science community to the people who can actually make the decisions that affect our lives, as Graham was saying? I don't know, Carmen, if you can comment on that anymore, but I would just comment that one EMBL alumnus, who I think everyone knows, Paul Nurse, was on the BBC radio this morning, and he was arguing strongly against, and he was just talking about the UK, but he was, I believe, the subtext that he was talking about is that the government are saying we are being guided by the scientists, but actually they're listening to a very narrow, selective. I don't even know how they selected them. They won't even tell us who these people are. But a very narrow spectrum of scientific opinion, not representative, I think, of the broad scientific view. So I don't know, Carmen, if you have any comment on that.
As I said, they don't tell us the names yet, so I cannot tell you exactly which is the committee. For sure, our science minister, he is, of course, meeting with different scientists in Spain. Most of them are closely related with coronavirus research. So we have a kind of a strong group in Madrid, actually, that are working with coronavirus since 30 years ago or so on. And they are meeting with them. They all come, for those of you that don't know, the Spanish Research Institute is kind of the public scientific organization in Spain, CSIC, that is where I'm working. It's 120 institutes all around Spain. So, of course, they come to us, let's say, to CSIC. And I can tell you that there is, internally, we created a platform at CSIC, which is somehow unifying around, at the moment, 200 groups that are doing or making research in different areas, because this is another problem. It's not a problem that you tackle in only one direction. You have many ways to take into account for covering this pandemic. So this platform that I'm telling, they are putting together the expertise of biologists, mathematicians, engineers, chemists, whatever, trying to give a global overview or in getting knowledge from all this data. So they are coming to this platform for sure. I know. The minister is coming there asking for questions or of course, telling people what we want to know. When the drugs are coming, which vaccines are we working in or whatever. But how direct is this relationship, or how much attention they pay to what the scientists are telling? This is very difficult, I guess, to quantify right now. I don't think...
So one comment I'd like to make, or an invitation to everyone, is what's impressed me in listening to all the people who've contributed from these three events we've held, is that the EMBL alumni community represent a very broad spectrum of highly informed political opinion that are now sampling opinion also from more than 30 or 40 different countries who have had people come and participate from essentially every continent. And my question is, how can we as a community then help to synthesize these viewpoints and present them in a way that we can communicate to our respective governments and hopefully attempt to influence the debates, whether through social media, whether through a white paper that could be sent to the European legislature or to our own national governments. So I'd like to open up for comment or discussion in the time remaining for anyone who would like to contribute. If you would like to put your hand up and contribute, Peter, perhaps you can help, or Noosh, you can help to select people. But if someone would like to contribute to this debate, or perhaps we can start off by having a vote. Do you feel, yes or no, that the EMBL alumni community can help to provide a better synthesis of the information available and help to communicate this both to the public and to our politicians? I'm going to vote yes. Can I bring in Keith Williamson, who had his hand up?
So I'm Keith Williamson. I worked at EMBL from 2001 to 2015. I'm not a scientist, although I do have O-level physics and chemistry. My profession is administration, in fact. So I think this discussion is getting, naturally enough, focused on science and what scientists can do with logic. And I think one of the things that I think the scientific community needs to be very careful about is overselling its importance in a case like this, because the pandemic is not only a scientific issue. Science is critical. We need something to stop people dying. But I saw a headline a couple of days ago where people were saying, I'm not frightened of COVID, it's not going to kill us, but we will die of starvation, which is the next threat that's coming through. And you need reasonably enough time to come up with analysis and demonstrations of cures or prevention. And you don't have time to do that using the scientific method. The politicians, on the other hand, and the people who are running the communities, actually need to know what to do now and how to come up with a quick answer which will mitigate the damage that's coming. And I think the other thing you need to recognize is that every branch of life has its fair share of idiots, even in the scientific field. And so, you need to be careful about saying all scientists should have input, because some of them clearly shouldn't. And I can tell the same story about accountants in the British Civil Service, where it was thought you should have accountants everywhere. And then I attended a meeting of accountants, and hearing some of my colleagues speaking, it was clear that accountants should be kept in cupboards a lot of the time. So I think you need not to take too much upon yourselves. I think what you're doing is great. I think it's interesting to see that you can look at the problems of curing the virus. The scientists who played most part in this are the epidemiologists, and their response, I think, has been well understood. We all know that we need to hide. We all know that we need to not to hurt other people, but at some time, we need to go out and buy food, and we need to look after our families, and we need to protect the environment. And I hate to sound like Mr. Trump, but, it does have a point. Is the cure going to be worse than the disease? And at some point, someone's going to have to say that some people are just going to have to die, because we can't all die. That's not worth it. So,
ultimately, we will all die. That's part of the normal life cycle. But actually, you raised a very good point, Keith. I think the general question about the role of science was also reflecting that the politicians are telling us that they are making their decisions, including unpalatable decisions about closing economies and restricting movement. And when we can ease the lockdown, they say they are being guided by the science and the scientists. And so it's a reasonable question for us as a scientific community to say, which scientists, because actually, Keith, you're completely correct in saying not all scientists agree on everything all of the time. And it's normal and right and proper that the science investigates all sorts of different options and possibilities, and then eventually over time, we coalesce around a conclusion that we agree upon based on a preponderance of data that's been thoroughly tested. And that's obviously very difficult. But in line with what Graham said, and you just said, I think perhaps a last vote for today, it would be interesting to just have the opinion of the community to say, if you don't mind me putting it this way, do you think, in this case, the strict lockdown measures the politicians throughout the world have taken in reacting to the epidemic, do you think the cure has been worse than the disease? Yes or no? But that's interesting, because I think we have a clear consensus emerging with some votes still coming in that there's a large majority of people supporting the view that the measures taken were appropriate. And I guess that's also a question that could be asked multiple times in the future to say at what point does that opinion change? Because presumably at some point we all have to go back to work.
And now over to Keith. I think he has something to say.
So just 2 points. The vote that you just had, Angus, was on the basis of whether the cure was worse than the disease. And you concluded that the measures taken so far were appropriate. Well, I don't think that follows the one from the other. And I would say, secondly, on that vote, following the scientific principle, the experiment isn't finished yet. We don't know what the downside is going to be economically. And saying that, you can't say now that we've been doing the right thing. I voted for worse, that the cure was worse than the disease. But I don't know anymore how anyone else does. We have to see how it develops. And I think when you have time to look back at all the reasons for the strange numbers that are coming out of different countries and why they occurred, you'll be very well placed to help governments when we get the next wave or next new virus to understand better how we should marry the competing priorities. And so this is an example where I think we're going a bit too far as scientists. Because if you follow your principles of experimentation, it's not over yet. That's all I wanted to say.
No, you're absolutely correct. And I thought I had actually said that at the start, that this is something where science will make the most complete contribution when it's able to look backwards at some point in the future. And yet, scientists, some scientists at least, are being consulted by our governments and asked to provide information. And so that's the difficulty, I guess, that we're in at the moment. But I completely agree with you that we don't have definitive answers. And these votes are for general interest. I don't think we'll be drawing any major conclusions from them. And it's true. One of the big complications I think we can all agree with is that it isn't just a question of how many people contract the virus or have a bad disease, but all the secondary and tertiary implications of, loss of jobs, psychological damage because of the indirect effects of the lockdown. These are absolutely critical issues as well that I think that we have to confront. And I certainly have no answers, but I think as a group, I think we are quite well placed to at least contribute effectively to the debate. And I would be very interested to see whether also working with members of your community, and I think Carmen gave a good start in this way, we can understand better how the voice of scientists broadly can be reflected in the public debate. Well,
Angus? Yeah.
Yes.
I have a question for Keith, because just to make things clear, I think there is no disagreement about the correctness of the policy of locking down society. I think initially when we saw the overflow of people coming into the hospitals and seeing the lack of capacity in the hospitals, etc. So during that period, I think you agree that locking down traffic and locking people up
was a right decision. I hope you agree with that.
I personally agree with that. I personally agree that because especially there was such a vacuum of information that you absolutely, I think, had to, I think they made the right decision to be cautious. I think the harder question is, as more data come in, to understand how do we reverse the lockdown, what's the trajectory, which groups of people. I think those are very difficult questions, and I don't have an answer. I do worry perhaps that the politicians have sort of got themselves into a minimum energy trap that's difficult to get out of. And I worry also about the effect of social media, because I think politicians are very sensitive to criticism on social media, and because that affects voter intentions. So I absolutely don't pretend to have answers to any of these questions. And I think as we've come to the end of an hour and a half on the discussion, I believe we have to draw it. But I think it's absolutely clear that there's a lot more things to discuss. And I'm optimistic that as a community, we can contribute very usefully to the future discussion.